Hello viewers, welcome to the course of Introductory Plant Pathology. We are dealing with the plant pathogens and fungi is our today's plant pathogen. Today we will discuss about the reproduction in fungi. As we all know that fungi have a very diverse array of reproductive strategies because there are diverse habitats in which fungi are growing, and there is also a great diversity in their genetic structures. So, looking to these diversity in the fungi, they have a typical type of reproductive strategy, and in that strategy there are three processes of reproduction in fungi. They are vegetative reproduction, asexual reproduction and sexual reproduction. Under vegetative reproduction, it is the type of reproduction which involves the somatic portion of the fungus thallus only. In vegetative reproduction, there are 7 classes of or types, we may say, of vegetative reproduction in fungi, that fragmentation, fission, budding through sclerotium, through rhizomorph, chlamydospores and oidium. Then we come to the fragmentation. In this process, the mycelium breaks into 2 or multiple of fragments, and they automatically become again the complete fragment and grows into a new mycelium. Then there is a budding. In the budding, the parent cell produces 1 or more projections called buds, which later develop necessary structures and detach to grow into new individuals, and budding is commonly seen in the unicellular forms like yeast. Then third is the fission. In this process, the parent cell splits into 2 equal halves, each of which develop into a new individual. Fusion is also very common in yeast. Then sclerotia. In some cases, as in the case of ergot disease of wheat, rye and bajra, we see the formation of sclerotia. Sclerotia are nothing but the complex structure of pseudoparenchymatous cells having so many types of spores and conidia within it. When it germinates, it gives out so many ascospores, and ascospores in due course of time infect the crop in the field. Then rhizomorph. In some higher fungi, several hyphae may become interwoven to form rope-like structure called rhizomorphs. Under favorable conditions, they resume growth to give rise to new mycelia. These type of structures are commonly seen in the mushroom and higher Basidiomycetes. As we have discussed just now, these are the pictorial presentation of the vegetative reproduction in fungi. The uppermost we are showing the budding, then fission, rhizomorph and the sclerotia. Then there comes the chlamydospores. These are thick walled intercalary cells which are a type of resting spores, and they also conserve the food material within them. Therefore, under unfavorable situations, chlamydospores behave like a resting spore for fungi, and on receiving the favorable conditions, they again germinate to form the mycelium or hypha. Then comes the oidia. These are the spore-like structures formed by the breaking up of the hypha cells. They do not store reserve food and hence cannot survive under unfavorable conditions. Such spores are produced in case of powdery mildew fungus Ericife. Then we see that asexual reproduction is exogenous as well as endogenous also. So, the spores or the reproductive units born at the tip or outside the vegetative structures are called conidia, and singularly we call them conidium. Two types of conidia are thylospores and conidiospores. The latter has got three types, that is blastospores, allurispores and phyllospores. The bearing structure of the conidia is called as conidiophore. Phore means the bearer, which bear the conidia above it. Generally, conidia are developed on a simple, that is without branched, tubular conidiophores. Some other types of conidia bearing structures are phylites. Phylites are nothing but simple a bottle like structures. Then cinema, coremia and acervulus, that is a heap like structure. Sporodokia, as we see in the case of fusarium. Pycnidia are there in the fungi imperfecti genera, and the sori. Sori is a very peculiar characteristics found in the rust fungi, that is the Puccinia. It is also known as singularly as sorus or pustule. Then we discuss about the endogenous asexual reproduction. The spores produced in sporangia, singularly known as sporangium. Sporangium is a name of a vessel or a container, and hence called sporangiospores, which are produced from the sporangia. Sporangiospores are of two types. First is zoospores or swarm spores, because they are having the flagella and they are motile in nature. Therefore, they are more primitive and aquatic type, and terrestrial fungi tend to produce zoospores, and they are found in the genus Echila and Saprolegnia. Second is the aplanospore, which are non-motile due to absence of the flagella. These are the different types of spores, as we have discussed, that vegetative hypha of the zoosporangium producing zoospores which are having flagella and they are motile in nature. Then sporangiophores are producing sporangiospores, and the conidiophores are producing conidia, oidiophores are producing oidia, and these are the chlamydospores, the resting spores, and then ascus is having the ascospores, and basidium is producing the basidiospores of higher Basidiomycetes. Then we come to the sexual reproduction. Sexual reproduction is known to occur in all groups of fungi except in the Deuteromycotina or fungi imperfecti, because the fungi imperfecti's perfect stages are present in the families of Ascomycetes and Basidiomycetes. Mostly it is in the Ascomycetes. It may involve fusion of gametes, gametangia or hyphae. The process may involve only fusion of cytoplasm, which is known as plasmogamy, or fusion of nuclei, which we call as karyogamy, or production of meiotic spores or meiospores. In most of the lower fungi, plasmogamy is immediately followed by karyogamy and meiosis. In higher fungi, karyogamy is often delayed, so that the hyphae remain dikaryotic. This phase of the fungal life cycle is called dikaryophase. Such fungi complete their life cycle in three phases: a haplophase, that is having single n of number, a dikaryophase having 2n number but not fused, and a diplophase where 2n number is there. Sexual fusion in fungi. Sexual fusion in fungi is of different types. First is the planogametic copulation. Motile gametes called planogametes undergo fusion. When both the gametes are motile and morphologically similar, the fusion process is called isogamy. Isogamy is commonly found in the Syncytium. When both the gametes are motile but different in the size, the fusion process is called anisogamy. This is also exemplified by Allomyces. And when one gamete male is smaller and motile and the other gamete female is larger and non-motile, the fusion process is called as heterogamy. This is the pictorial representation of the sexual fusion in fungi. First, we are seeing the planogametic copulation, where similar types of spores are fusing with each other is isogamy. Dissimilar types are fusing is known as anisogamy, and in oogamy there are one sperm and eggs are fusing with each other. In gametangial contact, we see especially that they produce the fertilization tube, and Intheridia fertilize the oogonia and the egg is formed. In gametangial copulation, in case of mucor and rhizopus, we see two different type of strains, that is plus strain and minus strain. They fuse together to make the female gametangium, and male gametangium make the diplophage. Somatogamy or anastomosis. Fusion occurs between two somatic cells and involves only plasmogamy. This results in the formation of dikaryotic hyphae. Hence, the process is called dikaryotization. For example, in the agaricus, it is very much present. Beside this reproduction of the fungi, there are two types of systems in the fungi. We see that there is a homothelism and heterothelism also. Based on the compatibility in sexual reproduction, the fungal hyphae can be distinguished into two types of homothelic and heterothelic strains. In homothelic forms, fusion occurs between the genetically similar strains or mating types. In such forms, meiosis results in the formation of genetically identical spores, whereas in the heterothelic forms, fusion occurs between the genetically different mating types or strains. The strains are genetically compatible and are designated as plus strain and minus strain. In such forms, meiosis results in the formation of both the strains in equal numbers. Then we see that what is the parasitism. The fungus is basically a saprophytic nature of organism, and as we know that an organism which lives on other organism without producing their own nutrient, then it is called as parasite. The relationship between a parasite and its host is known as parasitism. Many fungi grow on a non-living substrate. The organism of this type of mode of nutrition is called saprophyte. Based on the different types and modes of nature of nutrition, the relation between the host and parasite or saprophyte is termed in many ways as obligate parasite, that is known as biotroph, obligate saprophyte, facultative parasite, facultative saprophyte, hemibiotrophic and nectotrophic. Now, we come to the obligate parasite. Obligate parasites are those organisms which can only live in nature on a living plants. They are biotrophs. Facultative saprophytes are those group of fungi which can be cultured, otherwise they do not grow without any parasite, but they can be cultured on the nutritive media. Nectotroph are those pathogens that kill host cells in advance of its own growth and obtains nutrients from the dead host cells. Then hemibiotroph, parasite that requires living host cells for completing their part of life cycle is called as hemibiotroph. Facultative saprophytes are those organisms that normally lives as a parasite, but can also live as a saprophyte. Facultative parasites are those organisms that lives as a saprophyte, but can also live as a parasite. And saprophyte are those organisms that does not have any apparent interaction, beneficial or harmful, with any of the host. They only live on the organic materials. Now, we come to the nomenclature. In every plant and we see that there is a binomial nomenclature system, and just like that we do have this type of system in the fungi also, and naming of the fungi fall under the rule of International Code of Botanical Nomenclature. The scientific name of the organism follows the pattern of binomial nomenclature, which was first given by Linnaeus, which is composed of two words. Bi means two and nomen means the name. The first word designates the genus, and genus is always capitalized. The second word designates the species, and its name is not capitalized. When we come to the nomenclature, we have to have in mind that binomials were, when written, is underlined, and when printed they are italicized. Modification or updating, if any, is done only by the Committee of Fungal Nomenclature at each International Botanical Congress, which is held at every 4 years interval. Then we come to the life cycle of the pathogens. What is the life cycle? It is the sequence of the activities, or we may say the events which occur for completing the life cycle of a pathogen. We call disease cycle also, or the life cycle. Now, we want to study the life cycle of the Pythium. Pythium is mostly causing disease in the seedlings in nursery and in other plants which are growing in the pots. So, it is caused by the Pythium aphanidermatum, which I am showing today. Here the roots of the plants are first attacked by the fungal mycelium. Here then we see that in the asexual reproduction, the conidia are produced and they are liberate the zoospores, which shed their flagella and infect the roots of the seedlings in the pods. Then when we discuss the sexual reproduction, we see that there is antheridia and oogonia, and there is fusion, and they make the oospores, which is 2n in number, and oospore when germinate after the meiosis, they become n number of the spores, and then they infect the roots of the plants. This type of life cycle is there in the Pythium. Life cycle of Rhizopus and Mucor. In this type of life cycle, we see the asexual reproduction, in which mycelium is growing into the spores, which is n in number, and likewise they again repeat the cycle in the nature. But in during the sexual reproduction, the mycelium develops into two types of mating structures, that is the plus strain and the minus strain. These plus type and minus type of mating strains, they fuse and make the gametangia, which are haploid in nature. Then plasmogamy occurs and young zygosporangium or heterokaryotic zygosporium are formed. Then karyogamy is there, and zygosporangium after the reduction division gives out the 4 types of spores again to infect the plant. Then we come to the life cycle of Erysipeae, which normally cause powdery mildew in the plants. Powdery mildew is a very important subject, as they infect so many ornamental as well as commercially economically important crops in the field also. So, we see that here this is the leaf of grape infected with the powdery mildew fungus, and the asexual cycle is like this, that Clistothecia are produced on the leaves, shoots and berries of the fruits, and Clistothecia are formed. They germinate to produce the ascus, ascospore gave rise to the ascospores, and ascospores again infect the plant. Here in the sexual phase, the developing buds become infected with the powdery mildew fungus, and the fungus overwinters in the dormant buds here, and they infected bud give rise to young shoots. This is completely covered by the fungus. Then they give rise to the fungus sporulate on the surface, and they infect the plants. This is the life cycle of the loose smut of wheat. It is caused by Astellago nuda. It is a seed borne disease and very economically important, as after infection we see that only black powdery mass is there on the earhead of the or panicle of the wheat. So, as I told you that it is a seed borne nature of fungus, and the dormant mycelium is already present in the embryo of the seed. This invades the part of embryo seeds, and when we grow the seed in the field, mycelium invades young seedlings, and then mycelium follows the growing point of wheat plants and invades the grains at different sites. Then grain sites replaced by the masses of teleospores, which are just the black powdery masses, and at the site where the seeds are to be produced, we find only the black masses of teleospores, and this is the condition of the infected spikelet. Teleospores land on the flowers again by the rain or by air or through insects on the healthy plants and infect the again the developing grain. This is about the ergot of wheat caused by Claviceps purpurea. Ergot of wheat is very important disease, as it also causes the poisoning in the consumer of the product. When the ergot infected produce are being consumed by the human beings or by the livestock, the various types of toxic symptoms are developed in them, and sometimes it becomes fatal also. So, it is the life cycle of Claviceps purpurea. The sclerotia of the ergot fungus is grown along with the wheat seed in the field, and when sclerotia germinate, they produce the ascospores, and these ascospores then germinate the growing flowers in the panicles, and in asexually the honeydew, which is main symptom of the ergot disease, which is having the spores in it. The honeydew is a very sticky material and stick to the insect legs and other things, and when they visit the healthy plants, they also infect the healthy ones. Then stem rust of wheat, it is called by Puccinia graminis tritici. It is a very complex life cycle, because it completes the life cycle in two stages on two different plants. One is the wheat and the another is the berberis leaf. Actually, in India the wheat rust has become a great problem, because in the northern plains when we grow the wheat crop, they are always infected with the uridospores of rust fungus. Actually, it was a great enigma that from where these uridospores are coming from, and then the life cycle was completely studied, and it was found that it completes its life cycle on another alternate host, that is berberis plant, which grows on the hills, and at this time of growing of the wheat in a rabi season, from hills these uridospores are coming and infecting the wheat crop in the plains. So, how it completes the life cycle? This is the wheat crop, and uridia they infected with the rust fungus Puccinia. These are the uridospores. They after some time they become telutospores. When telutospore germinate, they gives the promycelium and some basidiospores are released. They infect at the hill region the alternate host berberis. On berberis, there is pycnidia formation and the formation of the plus and minus strains, and they fuse to form the acium. Then aciospores come to infect the wheat crop in the plains, and the uridospores are formed. Like this, it completes the life cycle on two different hosts. Hello viewers, today we have studied the introductory plant pathology and fungi as the plant pathogen. As we know that fungi is a very large group of organisms, and it has diverse habitats along with the genetic diversity, and therefore, it has so many reproductive strategies to reproduce and produce the cells of its kind. So, we have studied that there are three types of reproduction in fungi: vegetative, asexual and sexual reproduction. And we have seen the different types of spores helping in reproduction methods. Secondly, we have studied about the formation of spores and their motility, their non-motile nature, how they help in the reproductive system of the fungi. Then again we found that there is a different type of heterothelism and homothelism in the plants. About the binomial nomenclature, we have studied that how we name the genus and species in this fungi. Again we found that different types of life cycles of the plant pathogens which are infecting the plants and crop plants in the field.